Imagine evolving life, not over millions of years, but in a lab in just a few weeks. That's the idea behind directed evolution, a method that harnesses natural selection to engineer new enzymes. It's a process that won Frances Arnold the Nobel in 2018, and it's transforming how we make medicines, fuels, and materials.
Directed evolution. How we teach enzymes new tricks.
You might ask, why can't we just design better enzymes? Though we can predict enzyme structure with technology like AlphaFold, we don't know exactly which mutations will lead to a certain function. That's where directed evolution comes in. Instead of humans trying to predict the right answer, we let nature find it through guided trial and error. Evolution works through slow, random mutations and natural selection. Directed evolution takes those same principles and expedites it in the lab, compressing millions of years of trial and error into just a few experimental cycles. So, how does it work?
Scientists start by finding an enzyme that performs a similar function to the one they want. They then randomly mutate a specific part of the gene of that enzyme through techniques like error-prone PCR. The result is a library of millions of different enzyme variants. Next, these variants are expressed in hosts like E. coli or yeast. Each cell becomes a tiny test tube producing one variant of the enzyme. Scientists then screen or select these variants with a property they want, like faster catalysis or higher thermal stability. Screening usually involves using a selection pressure like cell death to quantify the best performers. The top variants are then sequenced and their genes become the parents for the next round of evolution, where more targeted mutations are introduced and the cycle repeats.
With each generation, the enzyme improves step by step, accumulating useful mutations. After just a few rounds, the final enzyme may look similar to the original, but have an entirely new function.
For example, Frances Arnold's group evolved cytochrome P450 variants to synthesize pharmaceutical precursors, performing carbene and nitrogen transfer reactions that no natural enzyme can do.
Other teams have evolved PETase enzymes that break down plastic bottles or lipases to stay active in industrial solvents. Even small changes can make a big difference. Improved enzymes can be 100 or even 1,000 times faster than the originals. So, what comes next? Scientists are now working to make this technique more precise and more generalized. They're using machine learning to find better starting points for evolution, incorporating metal cofactors, or even evolving entire metabolic pathways, expanding the scope of what we can do. Directed evolution has given us enzymes nature never imagined. Instead of calculating the perfect enzyme, we evolve towards it using biology's own search algorithm, refined by human selection. This technique has empowered scientists to make new enzymes that change how we make medicines, fuels, and materials, altering the very chemistry of our lives.